So today we are going to understand about Golgi apparatus. So first of all, Golgi apparatus was first discovered by Camillo Golgi. If you know about Camillo Golgi, he was a scientist who also discovered a method of staining that's called black reaction, in which he used a solution of silver nitrate and used it to stain individual nerve cell. This was a very easy method to see the cells by microscope in those days. Okay, so that's why he also won a Nobel Prize. So back on the topic, so let's discuss about the structure of Golgi apparatus. It is a membrane-bound cell organelle which has stacks of cisternae arranged parallelly, as you can see in the picture. So this is the picture, and you can see here cisternae are marked. All these structures, as you can see, are cisternae, and they are fluid filled. Okay, and they are also parallelly arranged over each other. Okay, stacking. You know for example, we stack clothes over each other. Similarly, they are stacked over each other parallelly. Now, it also has a close connection with the endoplasmic reticulum. You see in this picture. Here you can see the blue part, that is endoplasmic reticulum, and it is very closely related to endoplasmic reticulum because its main function is to modify and package and transport the substances that mainly come from endoplasmic reticulum. So you can see over here that blue vesicles that are coming from endoplasmic reticulum, that is basically proteins and lipids which come inside the Golgi apparatus, and Golgi apparatus main function is to modify, package and transport it to different parts of the cell, that is to the extracellular part and the intracellular part. So it just transport it to the extracellular part by exocytosis, and intracellular part by exocytosis also. The some parts also some vesicle formation also come from outside of the cell to the Golgi apparatus, and that's come by endocytosis. Okay, so here's a structure of yellow vesicle formed over here, that is lysosome. Actually, lysosome are not yellow. This is only a depiction here, and we will study about it later in the next class. Okay, so um, packaged and sent to the different organelles of the cell, the protein and lipids are required. The other function of Golgi apparatus is to make cell wall of our plant cell or animal cell. Actually, sorry, only plant cell, because animal cell do not have cell wall. It help its substance that are modified in small spherical structure called vesicle, as you have seen over here, the yellow things and pink things, and over here also the circular things actually are the vesicle in which the chemicals that are made by Golgi apparatus are stored. The chemical sugar molecule, lipid molecule that are modified are stored in vesicle. In plant cell, Golgi apparatus is simpler in the form and are called dictyosomes. So as you can see over here, there are many stacking over each other, but in plant cell, there is only one cistern. Okay, so this cistern is known as dictyosome. Also, they are not present in RBC of mature human body because the main function of RBC is to transport organ to the tissue. So, increase the amount of oxygen and increase the efficiency of the RBC. The RBC, as they get mature, they just degenerate all the organelles that are present in them. It also plays an important role in the formation of acrosome of the cell, sperm. Acrosome is the tip of the sperm, which you will study in class 12 biology chapter, that is human reproduction. Now let's go on the diagram again, and as you can see over here, that there are two parts over here. First is our cis phase, and second is our um, first is our cis phase, and over here it is trans phase. So the cis phase and trans phase are very important because the cis phase is the convex phase or formative phase. This cis phase, the vesicle come from the endoplasmic reticulum, and on the maturing phase or the trans trans phase of the apparatus, they are then dispatched to the intracellular and extracellular targets. As you can see over here, the intracellular targets, there are lysosomes, and extracellular target that are transported by exocytosis. The Trans phase is actually convex in structure. Now, summarizing the last part, that is, trans phase or maturing phase is concave, and cis phase or forming phase is convex. So, that's all for today. In this lecture, we have understood about Golgi apparatus, and in next lecture, we'll understand about lysosome. So, till then,